Okay.
Okay, great. And if you let me share my slides. Great. So it is my great pleasure today to introduce Julie Jung, who will be presenting her PhD research. Julie did her bachelor's degree in biology and environmental science at Williams College, and she has a long-standing interest in how animals use sensory information to assess risk and make behavioral decisions. As an undergraduate, she studied foraging decisions of chipmunks, which eavesdrop on bird alarm calls to assess predation risk. And she used acoustic playback experiments to test how road noise can change the chipmunk's behavior. Julie joined my lab in the summer of 2015 to study the hatching decisions of red-eyed tree frog embryos, which use egg vibrations to assess predation risk, again using playback experiments to test how aspects of development change the embryo's behavior. Julie's research is part of a collaborative project with vibrations engineer Greg McDaniel, who co-mentored key parts of her work. So Julie developed a skill set at the intersection of biology and vibrations engineering, which meaning that she can do a combination of things that neither Greg nor I alone can do. And one thing that Greg and I do together is to develop devices like this to ask questions about how embryos use vibrations. And in Julie's first field season, we finally got a new playback system working using eggs in trays, and Julie has made excellent use of it over 5 rainy seasons in Panama. Julie is an author on the first proof of concept paper using that playback system, and that took a long time to get published, probably due to the novelty of the approach. So we did a lot of work together to test and validate the playback system. And Julie is an author on a substantial methods chapter from that work, currently in press, which establishes new ways to study embryo behavior. Also in her first field season, Julie was part of a team project that determined that hatching ability is not what limits when embryos first show an escape hatching response to snake attacks. And that was her first publication from my lab, and the discovery that motivated some of the work that Julie did for her dissertation. So part of Julie's research examines how embryos use information from vibrations to assess risk, and it advances our understanding of both embryo cognition and how animals use incidental cues to make behavioral decisions. Julie also studied how embryos can sense vibrations in the first place. And 2 of her dissertation chapters documenting the role of 2 different mechanosensory systems that embryos use to assess risk were published as highlighted articles in the Journal of Experimental Biology. And this is really groundbreaking work. There are a lot of animals that hatch in response to egg vibrations, but before this, we didn't know how any of these animals, any of these embryos sense vibrations. And to figure this out, Julie had to do a lot of pilot experiments and methods development. And that kind of work is crucial for scientific progress, but it's really rarely evident in publications. We see what worked, but we don't always see the backstory of what it took to get there. Julie has been a great team member, and she has helped to mentor many other students, both in Panama and in Boston. She is an author on 18 conference talks and posters, some that she presented and others that her intern and undergraduate collaborators presented. She's also given 8 invited talks, including at Salisbury University and at Vassar. And as well as being an integrative biologist with a diverse skill set, Julie is a pretty hardcore climber. And I suspect that she applies some of the same personal strengths, resilience, and problem-solving skills to the challenges that she encounters in these very different parts of her life. When she leaves BU, Julie will be taking on another new challenge and learning a new set of methods, studying phenotypic plasticity in nematodes as a postdoc at the University of Utah. So I will stop sharing my slides and ask Julie to share her slides. And Great, thank you. Everybody, thank you so much for joining us. And I am delighted to present to you Julie Jung to tell us about her PhD research on developmental changes in vibration sensing and vibration cued hatching decisions in red-eyed tree frogs. If you have questions for Julie as you go along, as she goes along, please put them in the chat and she will address them at the end. And Julie, the floor is yours.
Thank you, Karen. Thank you, everyone, for being here. Welcome. Thanks for coming. As Karen mentioned, I'm here to talk to you today about my dissertation research on vibration cued hatching decisions in red-eyed tree frog embryos. So I'll just jump right into it. For animals, human and otherwise, life consists of a series of decisions. Whether to fight or flee, hunt or hide, flirt or forget about it, every day, organisms must make behavioral choices such as these in order to survive and reproduce. Resolving such dilemmas requires the ability to detect what's going on in the environment, process that information, compare the situation to past experiences, and behave in a way that's appropriate to the circumstances. Those abilities are in turn affected by development. So the key question that I'm interested in addressing in my dissertation is how and why does development change behavior? We know that behavior changes with development for at least two reasons. First, your developmental capabilities limit what behaviors you can engage in. Second, just because you can do something doesn't necessarily mean you should. So development might also affect the relative costs and benefits that underlie behavioral decisions. And the specific behavior that I'm going to focus on today is escape hatching in red-eyed tree frog embryos. Now, red-eyed tree frogs lay eggs on plants over ponds. And usually within 7 days, embryos will hatch and fall into the water where they will develop as tadpoles. But they can hatch prematurely to escape from egg predators. And that behavior is cued by vibrations in attacks. Here is a real-time video of a clutch of red-eyed tree frog embryos being attacked by a parrot snake that we named poison ivy. And this species of snake is one of 5 snake predators that we commonly see chomping on our eggs. And we see that clearly this snake is not being very sneaky. As she takes a bite and chews, she pushes around the eggs quite a bit, introducing a lot of incidental vibration cues. And the surrounding embryos, just like the one shown here, respond to the physical disturbance by hatching out into the water below. This clutch is 4.5 days old, so about 35% premature. And eventually, more than half of the clutch escaped and hatched out into the water. What's interesting is that this embryo escape hatching response changes drastically from oviposition to spontaneous hatching. The earliest that we've been able to induce hatching in these embryos is at 3 days post-oviposition in response to strong hypoxia cues or lack of oxygen from flooding. And so we know that by 3 days, embryos have the ability to hatch. However, we do not see the onset of predator-induced hatching until the following day. This means that there is a period of development between when hypoxia-induced hatching begins and predator-induced hatching begins, where embryos being attacked by snakes are clearly able to hatch, but don't and just get eaten. And up to 10% of eggs laid can be consumed during this period. After the onset of mechanosensory cued hatching, it quickly becomes harder and harder to handle eggs without inducing hatching. So clearly, development affects the hatching response, but how exactly does it change and why? Does it reflect further sensory development or decisions based on changing trade-offs? The first part of my dissertation looks at the first of these categories, behavioral capabilities. Since hatching ability is established well before the onset of predator-induced hatching, it clearly is not the sole constraint on escape hatching responses to predator attacks. So what is? Perhaps this delayed response in snake attacks could reflect changes in sensory ability to detect vibrations. So my first research question addresses embryonic sensory capabilities. How do embryos perceive vibrations in ovo? So in chapters 1 and 2, we examine both the inner ear and the lateral line as potential embryonic egg motion sensors. And both of these chapters have been published in the Journal of Experimental Biology. And in this talk, I actually won't present data from chapter 1, since chapter 2 also incorporates the inner ear hypothesis. I will, however, briefly touch on some additional projects in this presentation that didn't make it into the dissertation proper. So these take a closer look at embryo ears and examine whether and how ear development affects sensitivity to vibrations. And then the second part of my dissertation focuses more on hatching decisions. So now that the embryo has developed the ability to sense vibrations, how does it make the decision to hatch? From the perspective of the embryo, this is a really tricky decision because snakes are not the only thing shaking eggs in rainforests. In particular, rainstorms generate intense vibrations but pose no threat to eggs. Thus, embryos face this discrimination challenge. Since anything that shakes the clutch is shaking the same physical structure, it can be really tricky for an embryo to tell if it's actually in danger. So how do embryos assess risk in incidental cues? For my third chapter, I will look specifically at embryo use of complex temporal patterns in risk assessment. Finally, for my fourth chapter, I want to examine how embryos behave in the face of ambiguous cues. To answer all of our research questions, we worked in Gamboa, Panama at the Smithsonian Tropical Research Institute, and I'm so lucky to have been able to spend 5 amazing rainy seasons here. Every night after a heavy rain, red-eyed tree frogs come out to mate. And they leave these neat clutches of fertilized eggs on leaves overhanging our experimental pond. So the next day we can go out with buckets and collect these little jewels and bring them back to a nearby ambient lab to run our experiments. So first, in order to determine the mechanism for egg motion sensing in these embryos, we tested 2 different hypotheses. The vestibular system of the inner ear and the lateral line system are both well-known vertebrate motion detectors. Both systems depend critically on mechanosensory hair cells. We compared hatching responses for red-eyed tree frog embryos in which each of these systems was functioning and non-functioning. And in order to measure ear function, we used a behavioral indicator called the vestibular ocular reflex, or VOR. And to measure ear function with VOR, we used a portable tadpole rotator. And this is a really fun device that lets us stick a newly hatched tadpole in a small tube. And then the rotator smoothly rotates the tadpole at 15-degree increments so that we can take portraits of them in different positions. And from these photos, we can use a program called ImageJ to measure changes in eye angle with body rotation to calculate reflex strength. We can then plot eye angle versus body angle and then calculate the amplitude of the best fit lines. So the average of the amplitudes of the resulting curves for each eye indicates the magnitude of ear function in the developing embryo. So this is an example of individual embryos, both with and without ear function. And the animal without ear function showed no vestibular ocular reflex, while the animal with functioning ears had a high amplitude for its VOR curve. And we functionally did the same thing with respect to lateral line neuromasts. Instead of looking at embryos before and after ear function, we found a way to test hatching responses in embryos with both functional and non-functional lateral lines. And in order to measure lateral line function, we labeled hair cell bundles using a fluorophore diasp. And diasp temporarily dyes mechanosensory cells in the lateral line system orange, letting us see all functional neuromasts in live embryos. We also treated embryos with an antibiotic, gentamicin, to temporarily ablate lateral line function. Notably, gentamicin ablates lateral line system function, but has no effect on the vestibular system. So here we show VOR, so basically ear function on the y-axis, And then these gray lines connect points representing stage and age-matched siblings. And these red individuals have been chemically treated with gentamicin and so have non-functional lateral lines. And the black are our untreated control individuals. And we were really excited to see that this treatment had no effect on the amplitude of these VOR curves. And this made it possible to test embryos both with and without ear function and with and without lateral line function, and test whether one or both of these mechanisms lets embryos sense predator vibrations. And to test this, we looked to see if there's a difference in hatching response in embryos in each of these groups. To induce hatching, we pretended to be a snake by manually jiggling embryos and eggs with a blunt probe for 5 minutes total in intervals of 15 seconds. And we did this to 4 different groups of embryos before and after the onset of ear function, after vestibular function was well established, and close to spontaneous hatching. And to each of these 4 groups, we tested embryos both with and without lateral lines. So first, I want to look at young embryos just over 4 days old and just at the onset of their hatching ability. So these overlap in age, but only the green group has functioning ears. So our results show us that even before ears start working in this blue column, lateral line knockouts in red have a reduced hatching response, revealing that lateral line function precedes ear function and enables very early attack-induced hatching. And as embryos develop, both ear and lateral line function seem to improve the hatching response. Our oldest embryos all hatched, so we couldn't pick out any treatment differences in hatching responses. So we also examined latency to hatch as a potentially more sensitive indicator of lateral line contributions. We found that even these older 5-day-old embryos with really well-developed ears still use lateral line input to accelerate their response. Okay, so next, we wanted to know more about how the lateral line system develops over the period of plastic hatching. So we took pictures of more embryos stained with diasp so that their neuromasts were glowing. And then we labeled each line of neuromasts in both frontal and dorsal view. We found that the number of neuromasts in all 7 lines increased with developmental stage across the known period of mechanosensory cued hatching. So in fact, from the stage where cued hatching starts to where it becomes consistent, the number of neuromasts more than doubles. And here we see individual embryos sorted by whether or not they hatched in response to jiggling cues on the y-axis, and then the total number of neuromasts that they had on the x-axis, all colored by developmental stage. And there's a couple of interesting things to point out here. First, both stage and the number of neuromasts were significant predictors of hatching. And also, when embryos had fewer than a threshold number of neuromasts, none hatched, whereas embryos with more neuromasts often hatched. And we found that that threshold number of neuromasts was 247. And for some comparative context, flounder, tuna, grouper, and catfish all hatch with only 2 neuromasts. And red-eyed tree frogs hatch with 247 neuromasts. That is many more than described for any other species at hatching. And this suggests that precocious sensory development may facilitate early escape hatching in this species. So in summary, we found evidence that embryos can use both their ears and their lateral line systems to sense egg motion cues. And really small developmental increments in both of these sensory mechanisms drastically change their capabilities to respond to snake predator cues. The research that I'm going to show you next is not technically in my dissertation since I'm still writing it up, but it's related to my work on sensory development. And I'm just very excited about it, so I want to share a bit with you here. So for this next experiment, we manipulated developmental rate by rearing embryos under different thermal conditions. Warm conditions sped up development and cold conditions slowed development down. And the idea here was that if mechanosensory cued hatching and sensory ability just happened to develop at the same time, just by chance, warming and cooling the embryos could desynchronize that match. And this figure shows the developmental trajectory under our 3 thermal groups. So we clearly see that warm-treated embryos developed faster and cool-treated embryos developed slower relative to individuals raised under ambient conditions, shown in green. And across these 3 temperature treatments, and for each tested individual, we quantified the developmental onset of escape hatching responses to 2 different cue types. So to assess when embryos begin hatching in response to hypoxia, we submerged eggs in degassed hypoxic water. And then to assess the onset of hatching responses to physical disturbance cues, we jiggled embryos with a metal probe, just as we did in our lateral line experiment. And here we see the onset of both hypoxia cued hatching, shown with these hash marks, and mechanosensory cued hatching, shown in solid color, across all 3 temperature treatments. So this top plot shows these 2 responses in terms of chronological age, while the bottom shows these 2 responses in terms of developmental stage. And we see that temperature treatment had a significant effect on chronological age at response onset, but no significant effect on developmental stage at response onset. So in other words, warm-treated individuals started hatching in response to hypoxia and jiggling much earlier in time, sometimes even 2 full days earlier than cold-treated embryos. But the first embryos to hatch to each cue type were the same developmental stage, regardless of temperature treatment. So the first thing I asked was whether changing the developmental rate of embryos disrupts the ontogenetic alignment of ear function, measured with the vestibular ocular reflex, and the onset of mechanosensory cued hatching. And we found that in each thermal treatment, ear function, or VOR amplitude on the y-axis, appears concurrently with the onset of mechanosensory cued hatching on the x-axis, even across the really brief 3-hour period between no response and yes response. So even in cool-treated embryos, which developed really slowly in that 3-hour period, we still see a synchrony between increase in VOR and the onset of the mechanosensory cued response. And as expected, we saw more change in the hot treatment, but the responses were similarly aligned. So changing the developmental rate of embryos does not disrupt the ontogenetic alignment of ear function and the onset of mechanosensory cued hatching. And this further supports our hypothesis that sensory ability mediates predator-induced hatching in these embryos. Okay, so next I wanted to take a closer look at embryo ears and explore how their development affects sensitivity to disturbance cues. So in order to do this, I got to spend almost an entire summer dissecting out a ton of teeny tiny ears. So this is a pen tip for scale, and that is 1 ear. And I looked at ears in embryos just after they started hatching in response to stimulated snake cues, and then sibling embryos 3 hours prior to that, to the onset of that response. And we see that embryo ears develop a ton in just this short 3-hour time period. These are confocal images of whole mount ears just before and after the onset of hatching. And we see sensory hair cells labeled in green and their synaptic connections in red. And this is an even closer look at one of the vestibular sensory surfaces. And we found that both hair cells and synapses develop a lot over the onset of this response. After the onset of this response, the sensitivity of vibration cued hatching continues to increase. So this figure shows hatching responses across vibration amplitudes at 3 different ages. And if you compare the hatching responses of 4-day-old embryos, these this line in orange, versus 5-day-old embryos in light green, we can tell that older embryos respond to much lower amplitude vibrations, reflecting an 80-fold decrease in sensory thresholds to feel vibrations. And this growing sensitivity of vibration cued hatching in older embryos makes a lot of sense based on their continued ear development. So ears grow really rapidly in both size and complexity throughout this period of increased vibration sensitivity. So in summary, clearly developing sensory capabilities of both the vestibular and lateral line systems play a role in enabling predator-induced escape hatching. But again, just because an embryo can sense vibration and hatch does not necessarily mean that it should. So now I want to transition to the second part of my dissertation, which focuses on hatching decisions. And as a reminder, not everything shaking eggs in a rainforest is as threatening as a snake. For instance, rainstorms also generate intense vibrations in egg clutches, but they pose no threat to eggs. Evidently, there's a lot of uncertainty and ambiguity inherent in cues. So embryos face a discrimination challenge where it can be tricky to make an optimal hatching decision. Since cues can be tricky to interpret, embryos will sample multiple vibration properties to assess risk and distinguish among different types of physical disturbance. So for instance, we know from prior research that the duration of and intervals between vibrations matter, and low frequencies are stimulatory while high frequencies are inhibitory. And vibrations recorded in attacks and played back to embryos can induce hatching, and so can simple synthetic stimuli made from bouts of white noise. And we can create custom vibrations such that we have complete design control over properties like temporal pattern and frequency. We can then present these custom stimuli to red-eyed tree frog embryos in trays and track how much they hatch in response to really subtle differences in vibration properties. So as Karen alluded to, perfecting this system took a lot of time, but it ultimately gave us a lot of experimental freedom to tweak various stimulus parameters and test what precise properties these embryos care about when making their decision to hatch. So one of the vibration properties that embryos use to modulate escape hatching is frequency. So these gray bars show hatching in response to synthetic stimuli of different frequencies. And then in red is the mean spectrum from parrot snake attacks. And in blue, the spectrum of rain. And we see that embryos hatch much more in response to these low frequencies and less in response to high frequencies. And this is consistent with the fact that rain excites a lot of high frequencies, which indicate low risk and inhibit hatching responses. Embryos also use temporal pattern. So this shows a contour plot of hatching responses to playbacks varying in the duration of and interval between vibration pulses. And some patterns, like here in yellow, cause higher hatching than others. And we might expect that the properties of natural stimuli would match up with these embryo responses to synthetic stimuli. And in our initial analysis, the mean pattern of rain fell here, which does seem reasonable. But here is a cat-eyed snake, and here is a parrot snake. And it seems that their attacks would not induce hatching, but we know that they do, both in real attacks and in playbacks. So something seems to be missing here. And I think it suggests that just looking at temporal properties as these simple rhythmic patterns may be an oversimplification of risk cues. So perhaps embryos parse temporal patterns in more complex ways. Unlike synthetic vibrations, recorded snake attacks don't show consistent rhythms, and instead tend to include a mixture of shorter and longer pauses. So perhaps simplifying vibrations down to an average rhythm is obscuring some important information. Adult frogs are known to attend to multiple levels of temporal pattern information. Female tree frogs of many species discriminate between advertisement calls with different trill rates or pulse repetition rates within a single call. They also discriminate between calls that are produced at different rates. So a short interval between calls may make that male more attractive, but the same half-second interval placed within a call could make him unrecognizable. So the meaning of silence depends on where the gaps fall in relation to these other elements in a complex pattern. So perhaps embryos, like adults, also parse temporal pattern information across multiple levels of analysis. And to test this, we conducted a series of vibration playback experiments. And for each experiment, we constructed vibrational stimuli with different combinations of the same three simple elements: 0.5 second duration pulses, 1.5 second intervals, and 30-second long gaps. And based on the responses to rhythmic playbacks, we expect 0.5 seconds on and 1.5 seconds off this pattern to elicit high hatching, while combining the short pulse duration with 30-second long gaps should not. However, how do embryos respond to stimuli that combine all three elements in different ways? So all of these experiments were based around this simple rhythmic pattern of 0.5 second durations and 1.5 second intervals, which we already know elicits a lot of hatching. And we introduced a single 30-second gap after the first three pulses, creating a prefix to make a slightly more complex temporal pattern. And if the meaning of this long gap is consistent, it may reduce the response to this base pattern. However, since snake vibrations, as you saw in the video, can include longer gaps interspersed with shorter ones, we predicted that the presence of this long gap may heighten risk perception and amplify the hatching response. And our two controls featured each pattern component on its own. So either a single prefix or these repeated long gaps interspersed with single short pulses. And we found that the prefix alone and long gaps alone elicited almost no hatching. And this result is consistent with what we would predict based on our model of hatching responses to these rhythmic temporal patterns, right? That 30-second long intervals should yield very low hatching. But adding a single gap to a base pattern after a single 3-pulse prefix actually increased hatching rather than reducing it. And this suggests to us that even though long intervals are not scary to embryos in our simple analysis, as part of a more complex pattern, they can add to risk perception. So this might be relevant in a natural context as a way to help distinguish snake attacks, which have these long gaps, from hard rain, which tends not to. Okay. We then asked how much vibration is even necessary to elicit high hatching. So we tested smaller and smaller segments of the base pattern, so groups of 10 pulses and 3 pulses separated by these 30-second gaps, compared with our base stimulus as a control. And this bottom stimulus had only 30 pulses, while our base had 150 pulses. So we've replaced 80% of the vibration with silence. Does this reduce the hatching response? We found that the hatching response was similar to all three stimuli that included periods of the scary base rhythm, even periods as short as 3 pulses. However, if considered on a per pulse basis, so here we've normalized by the number of pulses in each stimulus, we see that hatching responses increase with the amount of silence relative to pulses. And in addition to hatching proportions, we also considered the timing of hatching. And we found that in real time, inserting long gaps slowed hatching. So embryos waited for longer to hatch. But when you subtract the times of the gaps, considering only periods of vibration, we see that embryos hatched with fewer vibrational pulses when they're interspersed with long gaps. And our data from this experiment suggests that some perception of risk is continuing to accumulate through these silent intervals. So the meaning of silence seems to be contextual and can actually elicit more and faster hatching in embryos. We saw that adding a 30-second gap increases the hatching response, which suggests that embryos are retaining information for at least 30 seconds without stimulation. But is there a point at which this no longer occurs? We tested gap lengths of 30 seconds, 45 seconds, and 60 seconds as compared to our base stimulus in order to look at this. And we found that gaps of 30 seconds and 45 seconds both significantly increase hatching compared to the base stimulus. But when the gap gets to be a minute long, the hatching response goes back down to what it was for the base stimulus. And the latency to hatch was around the same for all stimuli, regardless of gap length. So our results suggest that embryos have a mechanism to retain information over at least 45 seconds without vibrational stimulation. And this may not seem like long, but for comparison, the active time over which some adult frog females can remember which of these two males was making a more attractive call is not much over 45 seconds and oftentimes less. So our results suggest that the information processing and retention abilities of frog embryos, at least for certain critical types of information, is on par with those of adults. And we expect embryo behavior not to be as complex as the behavior of adults. But for some crucial behaviors important for survival, we may not be giving embryos enough credit. We show here that embryos, like adults, can parse complex temporal patterns on multiple levels and retain salient predator cue properties for long periods of time. All in all, embryos practice a very impressive, highly functioning decision-making process. So we knew that they use multiple vibration properties to distinguish between threatening and non-threatening stimuli. And this work demonstrates another level of complexity to embryo risk assessment strategies. And our study of complex temporal pattern processing in these embryos could motivate and inform a reanalysis of natural vibration recordings, utilizing more complex decision rules based on these identified embryo capabilities. So this might help resolve the behavioral mismatch in our initial analysis of hatching in response to these rhythmic temporal patterns. And hopefully, this will bring us one step closer in identifying the plausible analysis rules under which measured properties of snake vibrations would appear to elicit hatching and those of rain would not. Okay. So for my last chapter, I really wanted to examine the notion of uncertainty or ambiguity in cues. And the information from biotic and abiotic environments that animals use to inform their behavior is almost never perfect, right? Even stereotyped auditory or visual signals that are initially clear and honest because they've evolved to influence the behavior of others can change and degrade during transmission due to a gamut of natural and/or anthropogenic noises. Moreover, not all signals are honest, since animals can evolve to deceive each other. So a relatively large body of work addresses ambiguity in contexts such as dishonest signals or effects of environmental noise on communication. However, we understand considerably less about how animals cope with ambiguity in incidental cues generated by non-communicative activity. So examples of vibrational cues include frogs that fall silent in response to footfalls, elephants that pick up seismic vibrations through their feet, and antlion larvae that can detect the footsteps of an approaching ant. And such cues are critical for foraging and for defense, but selection on the cue producer might act to reduce their perceptibility. Thus, compared to stereotyped signals, incidental cues may be fundamentally more variable, ambiguous, or cryptic. So all of this is to say that animals face substantial variation in both signal and cue ambiguity in making decisions. So how do red-eyed tree frog embryos respond to vibrational stimuli that vary in ambiguity, and how do these responses change across development? So in assessing risk cues, embryos can either make the right decision to hatch when there's high risk or wait when there's low risk, or two types of mistakes. So miscues occur when embryos in danger fail to hatch, and false alarms occur when embryos hatch early unnecessarily. And the mortality costs of these embryos can change across development. So these younger, underdeveloped embryos face more challenges in the water and are less likely to survive aquatic predators. Thus, they face a higher cost of false alarms compared to these older embryos. So when presented with ambiguous cues, we predict older embryos to hatch more than younger ones, showing adaptive developmental change in how embryos make their hatching decisions. To test this hypothesis, we performed two experiments that varied ambiguity in different ways based on hatching data from prior studies. This first experiment varied ambiguity in the temporal pattern domain. With this clearly threatening in pink, this clearly threatening stimulus represents the tip of the hatching landscape, where yellow means more hatching. And then our second experiment varied ambiguity in the frequency domain, with similarly threatening low frequencies, benign high frequencies, and then in between ambiguous frequencies. And in the temporal pattern experiment, all three stimuli varied ambiguity in temporal pattern, but kept a consistent, constant, threatening low frequency. Whereas in the frequency experiment, all three stimuli varied ambiguity in frequency, but kept a constant, threatening temporal pattern. Thus, as an additional form of ambiguity in both this green stimulus or the clear cue of low risk, and then to some extent, this orange ambiguous cue, both frequency and temporal properties provided conflicting information. So this was true in both experiments with scary frequencies and non-scary temporal patterns, and scary temporal patterns and non-scary frequencies. So looking at the hatching responses in both experiments, we do see an interaction effect with the greatest developmental change in response to the more ambiguous stimulus in orange, consistent with our prediction of adaptive ontogeny. So younger embryos facing more risk in the water hatch less in response to ambiguous cues, while older embryos facing lower costs for false alarms opt to hatch even when faced with ambiguous information. So the fact that we found similar effects of ambiguity across these two very different cue property domains really strengthens the case for ontogenetic adaptation. And as we've seen already, the latency from stimulus onset to hatching is often a useful indicator of cue sampling. And when we take a look at how long it took for individuals to make their hatching decisions, in both experiments, as expected, younger embryos delayed hatching for longer than older embryos. And this suggests that older embryos generally take less time to assess cues and base their decision to hatch on less information. In general, we might expect to see inverse patterns of behavioral responses and latencies to respond, such that stimuli that elicit more hatching also elicit faster hatching. And we do see this pattern in our frequency experiment, where our threatening cue in pink yielded the most and also the fastest hatching, and our benign cue in green yielded the least hatching and also the slowest hatching. And then the developmental change in both variables was greatest in response to the most ambiguous stimulus in orange. However, latency patterns, unlike hatching responses differed between experiments that varied ambiguity in temporal pattern versus in frequency spectrum. So in the temporal pattern experiment, we didn't see the neat inverse responses like we saw in the frequency experiment. And we think this is because information from these vibration properties, temporal pattern and frequency, accrues at very different rates. So in general, information accrues at some decreasing function of sampling. But the rate depends on the type of cue and cue property. So some properties, such as frequency, these may be evident immediately. And in contrast, temporal property information accumulates gradually. So the more rapid accumulation of frequency information compared to temporal pattern information may explain patterns in our latency data. First, in the temporal pattern experiment, the cue conflict between frequency and temporal properties seems to decouple the linkage between hatching response and hatching speed. So in fact, for younger embryos, the fastest hatching occurred with the benign pattern that elicited the least hatching, this dot in green. And this pattern had the longest duration of vibration pulses, and thus exposed embryos to a rapid accumulation of vibration at frequencies indicating high risk, but a slower accumulation of temporal pattern information indicating low risk. So in this situation, individuals perceiving lower false alarm costs, like older embryos closer to spontaneous hatching, may cross a threshold to hatch based on scary frequency cues before sufficient temporal information accrues to lower their risk assessment. So we've shown here that latency can reveal nuances of how embryos deal with conflicting information from cue properties. To recap, this study shows that embryos adjust hatching decisions to ambiguous cues in predictably adaptive ways. And we hope our conclusions here may inform approaches to studying how other animals use incidental or ambiguous vibrational cues to make important behavioral decisions. In summary, this dissertation advances our understanding of the sensory mechanisms and cue assessment strategies underlying the vibration-cued hatching of red-eyed tree frogs, with broader implications for understanding the development of behavior. So first, we demonstrated that both embryo ears and lateral line systems play important roles in mediating the hatching response. Then we examined how ear development is correlated with improved sensitivity thresholds to vibrations. Lots of embryos across diverse animal taxa, including fish, amphibians, reptiles, and invertebrates, commonly use physical disturbance cues to inform their hatching timing. So here I've showed photos of just a few of my favorite examples. Despite widespread occurrence of mechanosensory cued hatching, until now, the mechanism for mechanosensing in ova was entirely unknown. I think that it would be interesting to investigate what sensors are involved in these widespread responses. And Chapter 3 addresses the very general and understudied issue of how animals parse temporal pattern information from incidental cues. We found that red-eyed tree frog embryos perceive temporal patterns of vibrations at more than just one scale. Finally, we show that embryos make decisions in a way that matches the changing balance of costs and benefits. And this contributes to the body of evidence for adaptive ontogeny and to our more general understanding of the multiplicity of factors affecting behavioral development. Collectively, my dissertation research shows that embryos combine information from multiple sensory modalities and deploy impressive cost-benefit risk assessment to determine whether or not to hatch. My work on decision rules highlights the potential nuance and complexity of cue assessment, even in early life stages. It cautions against oversimplifying assumptions about early life stages and demonstrates the value of embryo hatching behavior in research in animal cognition. So I now want to take this chance to thank all of the people who have made this work possible. So first and foremost, I'd like to thank my advisor, Karen Workington. My partner likes to joke that Karen is like a Karen for me. cairns are these small stacks of stones constructed along a sometimes very subtle trail. And as climbers, we like to use them as landmarks to guide our way and keep us from getting lost. And I think this analogy is apt to say the least. Every time I have felt stuck or lost in research or even in life, I could always count on Karen to prop me back up and help me get back on track. I also want to thank our engineering collaborator and my co-advisor, Greg McDaniel, for his vibrometry expertise, as well as several other key collaborators and co-authors. I thank my lab family, past and present, for the hammock hangouts, the birthday cakes, the aguardiente, the happy hugs and the sad, just all the love, the friendship, the logistical and emotional support throughout the years. I could never have made it here without you. Also, thank you to my partner, Tom, and our pup, Luna, for always cheering me on and bringing me tea and generally just making me very happy. And finally, thank you to my family. I can credit everything good in my life to my loving Amma and Appa, who rededicated and rearranged their entire lives to facilitate the success of their children. And also my older brother, my Oppa, who spent much of his childhood trying to shake his little sister off of his leg, and then much of his adult life trying to make sure she is safe, supported, and happy. One more slide. So this dissertation has truly been a collective effort. So I lastly want to thank my dissertation committee for their key input and advice, and to you all for sharing the virtual table with me today. That's all I have. Thank you.
Woo!
So we'll have a chance now for public questions for Julie while we're all here together before we go to the committee defense. So please feel free to put a question in the chat for Julie, or to raise your hand and we'll call on you to unmute yourself and ask her questions. And for those of you who are joining us from other places, I'm just gonna put a link in the chat that if you want to give me your email address, we will also let you know after the private part of the defense is over. That we'll be doing next. But any questions, public questions for Julie?
Yes, Pete.
Yeah, no, fantastic job, Julie. I just, as we were going, I've seen the whole thing, but as you were going through, I was the bit where the response to hatching drops off after 60 seconds and it peaks at 45 seconds. I was just curious if from the recordings you have on snakes, whether you have some sort of distribution of the pauses in snake attacks to know how long those pauses are in snake attacks. Do we know that, or is that what you alluded to as sort of the next step that you're going to get to next?
I think it is a plausible next step. We have a ton of recordings. That's why I think a nice step would be just to look at those recordings and make use of them. There's a lot of range from just kind of visually looking at them. There's a big range, even among the species and among, like, I know that they tend to kind of eat in different ways. I think some species are like more aggressive eaters, and so maybe that would change their temporal patterns. In terms of like, there is a paper that did look, so Karen's 2007 paper that did take a look at recordings, And that's where we got those kind of mean values from.
Okay.
So yes, there is some work that has been done. There's a lot more that could be done in order to analyze those recordings.
So those mean values are the ones you were using in the simple pulse
Yeah.
pause. So they were less than 45 seconds, right?
Yeah, as a mean. But again, that kind of obscures a lot of information. What does that even mean?
Yeah, cool. Thanks.
Thanks everyone for coming. Hope you enjoyed.
Any other questions for Julie? And her committee will have plenty of questions for her in a few minutes, but we'll give others a chance first. If you. Okay, I'm seeing a bunch of comments coming into the chat. If you don't have questions and you just want to
Say hi.
Say hi or anything, feel free to put in the chat. We can look at that.
I have another question if nobody has another question. I'm just curious, and we can talk about this later, but the postdoc that you're going to, you said it's on nematodes?
Yes.
Can you tell me a bit more about it?
Yeah, it's on nematodes. It's for like so Karen's phenotypic plasticity class is gonna come. Oh, there's people coming into the wing. Is gonna come in handy for this. So this lab is looking at these different mouth forms.
Okay.
And so it's also kind of developmental plasticity, but in this whole other system.
Okay.
And what's nice about it is that it's like a sister species to C. elegans. So there's a lot more kind of genomic tools.
Genomic.
Mm-hmm.
Cool. So you're going to be going down new avenues there as well.
Yes, but still a really cool example of developmental plasticity.
Yeah, okay. Very cool.
Yay.
Okay, well, if there are no other questions, let's thank Julie again for her great presentation. If you are not on the BU email, biology email list, and you want an email in a while after the committee deliberations, put your email in that document for us. And thank you all for coming. And the committee will now, I guess, will adjourn to the separate link to talk with Julie there.
Karen, can you remind me, was the separate link in an email from you or from Julie?
Separate link in email from me.
Thank you.
Do we just stay here, Karen? I mean, we probably could just stay, no?
Do you want to just say, is that, I guess, that could work too, if
Okay.
just wait for other people to leave. Is that better?
Sure.
Okay, we'll do that.
I'll stop recording.